Welcome to the Deep Dive. Today we're diving deep into a topic that's really transforming healthcare and research, enterprise-level genomics data processing. You've shared with us this comprehensive blueprint, enterprise genomics data processing pipeline architecture, implementation operations by Sri Sivakumar Ramar. And wow, this document looks like a goldmine for understanding how, massive amounts of genetic information are securely and efficiently managed at scale. So our mission for this deep dive is pretty clear. Unpack this blueprint. We want to trace the journey of genomic data, from those raw sequences right through to clinical insights. And it all seems to flow through this sophisticated system built on Databricks. We'll aim to reveal the critical layers of processing, the paramount importance of security, and really what makes these pipelines so powerful for everything from research to patient care.
Yeah, what's really striking about this document, I think, is that it moves beyond just theory. It's not just a high-level vision. It actually provides practical, ready-to-implement architectural patterns. It feels like a genuine guide for building a robust system. And we're talking about handling some of the most sensitive, most complex data imaginable here.
Exactly. So if you're looking to quickly grasp the essentials, the nuts and bolts of large-scale genomics data management, well, you're about to get a crystal clear picture of that whole journey, from raw DNA reads all the way to actionable insights, and crucially, within a secure and scalable framework. Okay, let's start at the beginning then. What's the core objective? What's guiding this entire blueprint? It seems to be all about designing and implementing a scalable, secure, and compliant genomics data processing pipeline using Databricks.
Yeah.
So this isn't just about shuffling data around, is it?
No, not at all. It's about establishing a really meticulous, trustworthy pathway for genetic information. And if we, zoom out to the bigger picture, the fundamental goal is enabling rapid, reliable analysis of truly vast genomic data sets. The blueprint lays out precisely what this pipeline aims to achieve. It covers everything from ingesting raw genomic data, typically those FASTQ or BAM files you mentioned.
Right, the raw reads and the aligned ones.
Exactly. All the way through performing these intricate steps like alignment, variant calling, annotation, then storing that process data in Delta Lake, governing access with Unity Catalog, ensuring robust audit logging, and importantly, supporting downstream analytics, machine learning, and automating deployment for reproducibility. It's quite comprehensive.
It really sounds like it. And the data itself, you mentioned it's incredibly rich, packed with highly specific information. Can you walk us through some of those key data points? Why are they so crucial for this kind of analysis?
Absolutely. Yeah, when you're dealing with genomic data, every piece matters. You've got things like sample ID, obviously, for unique identification, then chromosome and position to pinpoint the exact location on the genome, and reference allele and alternate allele, those show the actual genetic variations compared to a standard reference.
Okay, that makes sense. The basic coordinates and the variation.
Right. But then you also have these critical quality metrics like QoLL, which is essentially a quality score for the variant call itself, DP or read depth that tells you how many times that specific spot was sequenced, which impacts confidence.
Ah, okay. More reads, more confidence.
Precisely. And AF allele frequency, which tells us how common a variant might be in a population. But, the real challenge and where the value often lies is translating these raw genetic variations into meaningful insights. Biological or clinical insights.
That's the key step, isn't it?
It really is.
Yeah.
And that's where columns like consequence come in. It predicts the functional impact of a variant and gene, the specific gene symbol affected, plus ClinVar significance. This provides that crucial clinical interpretation like pathogenic or benign.
Wow.
Yeah, the example data in the blueprint really brings this home. It shows things like a BRCA1 gene with a pathogenic missense variant, or maybe TP53 with a benign synonymous variant. It highlights the kind of critical, potentially life-impacting information this pipeline is built to handle.
That really underscores the sensitivity and importance.
Oh.
Okay, so we have the objective. We understand the richness of the data. What's the next step in actually building this, laying the groundwork, setting up the environment? How does the blueprint guide that?
Well, it starts practically enough with creating dedicated workspace folders inside the Databricks file system or DBFS, things like MN Genomics SPRA, MN Genomics Process, MN Genomics Reference, just organizing the space.
Simple but necessary.
Exactly. And what's particularly well addressed is the ingestion workflow. For those initial raw FASTQ files, these files usually come straight from the sequencers, typically delivered to cloud storage buckets like Azure Data Lake or maybe AWS S3.
Right, they land in the cloud first.
Correct. And the blueprint shows how to securely mount these cloud locations within Databricks. It even emphasizes using things like OAuth authentication with client IDs and secrets to protect that incredibly sensitive data right from the get-go. Security is baked in early.
Good to hear. Okay, environment setup, data ingested securely. Now, this is where the heavy lifting happens, right? The processing pipelines. This feels like where the raw data gets transformed into something usable.
Absolutely. This phase is where the blueprint really shows its ingenuity, orchestrating these complex, often resource-intensive genomic analysis tools, all within that scalable Databricks framework.
So how does it structure these crucial stages? Let's start with alignment, taking those raw reads and making sense of them.
Okay. Yeah, the blueprint breaks this down into 3 main processing steps. Each uses specific, powerful genomic tools. First up is alignment. Here, it suggests using tools like BWA, Burroughs-Wheeler Aligner. Its job is to take those raw FASTQ files and map them or align them to a reference genome, usually a file like reference.fasta.
So like putting the puzzle pieces onto the picture on the box?
That's a great analogy, yes. It finds where each short read fits onto the long reference sequence. And the output of this step is typically an aligned BAM file, binary alignment map format.
Okay, when it's aligned, then the blueprint moves to variant calling, you said.
Correct. Step 2, variant calling. For this, it highlights tools like the GATK haplotype caller. GATK stands for Genome Analysis Toolkit, very widely used. This is the step where the pipeline identifies the specific differences, the variations between the individual's genome sample and that reference genome we just aligned to.
Finding the typos, essentially.
Kind of, yes.
Yeah.
Finding where the sample sequence diverges from the standard reference. And the output here is usually a VCF file variant call format.
VCF.
This VCF file basically lists all those identified genetic variations in a standardized way. Very important format in genomics.
Okay, so we have the list of variations, but they don't mean much on their own, right?
Yeah.
That brings us to annotation.
Precisely. The third key step, annotation. Here, the blueprint mentions tools like SnpEff. Annotation adds that crucial biological context to the variants found in the VCF file. It tells you things like, what gene does this variation fall into? What's its predicted functional impact? Does it change an amino acid in a protein, for instance?
Ah, so this is where we get the potential meaning, like pathogenic or benign potentially comes from this stage.
Exactly. It layers on that interpretive information, super vital for making sense of the raw genetic code. And critically, the blueprint points out that after each of these processing steps, the data, especially the VCF data, gets converted and stored into Delta Lake tables.
Okay, Delta Lake. Why is that important here?
Delta Lake is key for several reasons in this context. It provides a robust, structured storage layer right on top of your existing cloud data lake storage. It brings features like assay transactions, atomicity, consistency, isolation, durability, which is huge for data integrity.
Right, ensuring data changes are reliable.
Yes. And it enables schema enforcement, making sure your data structure stays consistent. Plus, crucially, for genomics and regulated environments, it provides data versioning. You can track changes, revert if needed, audit historical data states.
That sounds absolutely critical for this kind of sensitive, evolving data.
It really is. For integrity, for auditability, for reproducibility, Delta Lake is a cornerstone of this architecture.
Okay, so we had this massive amount of incredibly sensitive data flowing through these complex steps, landing in robust Delta Lake tables.
Yeah.
But how do you manage access? Keep it secure, keep it compliant, especially in a large enterprise setting. The blueprint mentions governance and security.
Yes, and this is where Databricks Unity Catalog comes into play. It's presented as absolutely central to the governance strategy.
Unity Catalog, what does it do?
Think of it as a unified governance layer for all your data assets on Databricks. It allows you to define external locations that securely link to your cloud storage where the data actually lives. And then on top of that, you define schemas like a genomic schema and tables like the annotated variance table we discussed. These point to the data in Delta Lake. But Unity Catalog becomes the single place where you manage metadata, lineage, and, importantly, access control for all of it.
Ah, a single pane of glass for governance.
Exactly. It really streamlines what can otherwise be a very complex security landscape, especially with data spread across different locations or formats potentially.
And what about controlling who can see what? That granularity seems vital.
Absolutely. And Unity Catalog excels there. The blueprint shows examples of granting permissions with fine-grained control. For instance, you could grant select on schema genomics to genomics readers. So a group of users can only read from that schema. Or you could grant all privileges on table annotated variants to genomics admins. giving administrators full control over that specific crucial table.
That level of control is really impressive. And necessary, I imagine.
Totally necessary. It's crucial for ensuring only authorized users can access specific slices of this highly sensitive data. Least privilege access, essentially.
This brings up a really important point you touched on earlier, compliance. Regulations like GDPR, IPEA are mentioned. How does the blueprint ensure you meet those requirements?
Right, that's under the non-functional requirements, and it's tackled head-on with auditing and monitoring. The blueprint details how to enable audit logs within the Databricks Workspace admin console. That's step one, make sure logging is on.
Capturing who did what when.
Precisely. Then it talks about setting retention policies for those logs, say keeping them for 1 year, which might be a compliance requirement. And it even shows examples of how you might process these logs later, like filtering for the Unity catalog service name to specifically see who accessed which tables or schemas and when. Provides an undeniable audit trail.
That traceability is key for compliance.
Absolutely essential.
And beyond just tracking data access, what about keeping tabs on the pipeline itself, making sure the jobs run correctly? Job monitoring.
Yes, that's covered too. It's about operational robustness and efficiency. The blueprint highlights implementing alerts, maybe based on retry policies if a job fails. Setting up notifications, perhaps to Slack or email, so the operations team knows the status of critical pipeline runs.
Proactive monitoring.
Exactly. And crucially, for cost management, it stresses enabling cluster auto-termination. Genomics workloads can be computationally expensive, so you don't want clusters sitting idle and racking up costs. Auto-termination spins them down when they're not needed.
Smart. These operational details really make it feel enterprise-ready, not just a proof of concept.
That's the goal, yes. Bridging the gap between possibility and production readiness.
Okay, so the data is processed, it's secure, governed, monitored. What's next? The blueprint doesn't just stop there, does it? It mentions integrating machine learning.
Right. And this is where, arguably, the true value of having this integrated pipeline really shines. Unlock the insights. By leveraging tools available within the Databricks ecosystem, like PySpark.ml.classification, you can train machine learning models directly on that processed, annotated genomic variant data stored in Delta Lake.
Directly on the governed data.
Directly on the governed data. The blueprint gives an example using random forest classifier. It shows how you can use tools like vector assembler to create feature sets from those metrics we discussed earlier: QOL score, DP, read depth, AF, allele frequency.
Using the data's own features to predict something else.
Exactly. It demonstrates building an ML pipeline within Spark to train, say, a variant classifier model. Think about the possibilities. This could be used for automated insights, like predicting the potential pathogenicity of a newly discovered variant based on its characteristics.
Wow, that's moving beyond just reporting variants to actively interpreting them at scale.
Precisely. It's a fundamental shift from purely retrospective analysis to potentially proactive data-driven insights, and doing it at a scale that wasn't feasible before these integrated platforms.
That's incredibly powerful. But making this whole complex system, ingestion, processing, ML, repeatable and reliable must be a challenge. How does deployment automation fit in?
Yeah, consistency and reliability are key for any enterprise system. The blueprint addresses this by building in deployment automation. It suggests using infrastructure as code tools like Terraform. With Terraform, you can define your Databricks jobs, clusters, permissions, everything in configuration files.
So you can version control your infrastructure and deployments?
Exactly. You version it, you can review changes, you can deploy consistently across different environments, development, staging, production. Alternatively, it mentions leveraging the Databricks CLI, command line interface, for scripting deployments. Either way, the goal is automation. Ensure deployments are consistent, reproducible, and drastically reduce the potential for manual errors.
Makes sense. Automation is key at this scale.
Absolutely. And if you connect this automation back to those non-functional requirements mentioned earlier, things like the ability to process thousands of genomes per batch, that's scalability, achieving less than, say, 6 hours per sample for variant calling, that's performance, ensuring encryption at rest and in transit for all data, that's security.
Right.
It's the combination of the architecture, the automation, and meeting these stringent requirements that truly makes it an enterprise-grade solution. The blueprint even includes pointers towards having operational playbooks for job monitoring, cluster management, periodic data access reviews, incident response. That speaks volumes about its real-world focus.
It sounds very thorough, but no system is without challenges. Does the blueprint acknowledge potential problems? What about risks and mitigations?
It does, which is good to see. It takes a practical view. It explicitly calls out risks like potential sensitive data exposure.
A major concern, obviously.
Huge. And the mitigation pointed to is leveraging Unity Catalog's fine-grained access controls, coupled with those robust audit logs we discussed, defense in depth. It also addresses the risk of cost overruns, especially with cloud compute. The mitigations there are using cluster policies to enforce limits and that cluster auto-termination feature to avoid paying for idle resources.
Practical cost control.
And pipeline failures. What happens when a step breaks? Mitigated by designing the pipeline modularly, implementing sensible retry policies for transient errors, and having comprehensive alerting in place so someone knows there's an issue.
That seems like a solid approach to operational risks.
Indeed. And there's another critical risk it points out, particularly relevant to science: non-reproducible results.
Yes. If you can't reproduce it, is it really science?
That's the core issue in many fields, and genomics is no exception. Changes in software versions, reference genomes, parameters, they can all affect the results. The blueprint mitigates this by emphasizing the use of version reference genomes and pipeline definitions, so you always know exactly which version of the reference, which version of the analysis pipeline script, which tool versions were used for a specific result.
Locking everything down for reproducibility.
Exactly. That commitment to versioning and documentation is fundamental for scientific rigor and ensuring findings can be verified and replicated later.
It really sounds like the author intended this to be, as they say, an end-to-end blueprint. It's got the architecture, the real code patterns or examples, the built-in governance and security, the monitoring, the deployment. It feels very complete.
I agree. It aims to be a comprehensive guide not just for building these systems, but for operating them successfully in a demanding enterprise or research context.
Well, we've certainly taken a comprehensive deep dive into this incredible blueprint for enterprise genomics data processing. We've journeyed from those initial raw files, the FASTQ reads coming off the sequencers, through alignment, variant calling, annotation, landing in secure, governed delta lake tables, and then seeing how that data can drive advanced machine learning models, all while being secured by things like Unity Catalog and robust auditing. You really get a sense of how this complex genetic information can be managed at an immense scale.
Yeah, what's truly impressive, I think, is how this detailed architecture manages to balance two critical needs. On one hand, handling the sheer volume and complexity of genomic data, and on the other hand, meticulously prioritizing the integrity, the security, and the reproducibility that are just non-negotiable for serious scientific discovery and clinical application. It's really a testament to how crucial robust data engineering and platform choices are. They truly underpin the breakthroughs we're seeing in genomics.
So what does all this mean for you, the listener? Well, it means that behind those exciting headlines about new genetic discoveries or personalized medicine, there's often an incredibly intricate, secure, and highly automated system like the one described here, making it all possible. The plumbing matters. And for you, thinking ahead, as genomic data becomes even more central, even more prevalent in healthcare and beyond, consider this. How might these types of highly automated, secure pipelines further democratize access to personalized health insights, make them more widely available? And, perhaps just as importantly, what new ethical considerations or societal questions might arise as this kind of widespread, efficient, large-scale processing of our most personal data becomes the norm? Something to think about.